Mitochondria are not like other organelles, especially in relationship to the cell. Mitochondria make their own ATP. They give the cell its energy. But how did the mito end up like this? One theory comes from Lynn Margulis. One theory that went over well was the endosymbiotic relationship with the cell. At first, there was a cell who needed ATP, who met with the mitochondria living free. The cell took it up through endocytosis. They lived happily in symbiosis. To allow the mito to make energy well, it gets its proteins from the cell. To get there, three things need to happen, I'm told. Bind to the membrane, pass through, then fold. Protein finds the mito through random motion.
But the mito's so small, that's a crazy notion.
Here's one thing you might find bizarre, but the mito's usually shaped like the rough VR. With that much surface area, it's easy to see how the protein can find the mito randomly. If it wants in, it better not fold. It needs HSC 70 to grab a hold. But it won't get through the membranes as such. A signal sequence is the final touch. The protein finds a signal acceptor. In this case, it's the Tom receptor. The protein binds to Tom, but what's more, Tom binds again to his friend Tom Poor. Tom Poor lines up with Tim 23. Now let me explain. Tim lets the protein through the inner membrane. The protein's pulled through the mito by and large by the signal sequence's disulfide charge. To stay unfolded, the protein can't do it alone. They need the help of molecular chaperones. Tim 44 recruits HSC, then transport happens super smoothly. Once the protein is in the matrix, a peptidase cuts the signal sequence. Now the protein folds when they help make the mitochondria run.